packaging steps and experience summary of lentivirus. Abstract. Lentivirus is a one of retrovirus. It can introduce target gene into some difficult-transfected cells, such as primary cells, and integrate target genes randomly into the host genome, thereby greatly increasing the transfection efficiency, with stable expression in cell lines for several generations. It can also be used for screening stable cell strain. Because there is a random integration with uncertain factors, some companies provide targeted integration technology to integrate target gene into the genome of the specific position, so as to ensure the high expression and not to generate damage to cell produced by random integration. Content. The process is that lentiviral vectors contain packaging, transfection and stable genetic information integration. Lentiviral packaging plasmids can provide all auxiliary proteins needed in transcription, packaging and recombination. In order to produce virus particle with high titer, it needs to use the expression vector and packaging plasmids to co-transfect cells, and do virus packaging in cells. The purified virus particles are secreted into the extracellular medium, and the supernatant can be directly used for host cell infection after centrifugation. Content. Lentivirus genome is a positive-stranded RNA. When the genome goes into cells, it is transformed into DNA by reverse transcriptosome by itself and forms pre-integration complex to enter the nucleus. DNA is integrated into the cell genome. The integrated DNA transfers RNA and returns to the cytoplasm, expressing target protein or producing RNA interference. Content. The lentivirus packaging system consists of a packaging plasmid mixture mix and a lentiviral vector plasmid. The vector contains the basic components of HIV, such as 5-LTR, 3-LTR and other auxiliary components. Contents. Contents. Packaging plasmid mixtures are not the same in different systems. The 3-plasmid system in the laboratory is taken as an example. The plasmid mixtures contain PMDL, VSVG, PRSVREV, and the ratio in 5:3:2. Among them, PMDL contains the GAG gene encoding the major structural protein of HIV virus and the POL gene encoding the virus-specific enzyme. PRSVREV has a regulatory factor REV gene that regulates the expression of GAG and POL genes. VSVG contains the VSVG gene that comes from the herpes simplex virus needed for viral packaging. Preparation. 1. Part 1. Reagent Chapter. Plasmid. 293 T cells with exponential growth. Virus packaging plasmid mix. 1 microsine G divided by microsine L mix equals PMDL, VSVG, Rev equals 5:3:2. Packaging virus plasmid is different by different vector systems. This system can be used for packaging BOBI, PILCO and PLV plasmid. Transfection reagents. Turbofector Lipo 2000. Reagent Chapter. Cells were collected by trypsin digestion, and cells were cultured on the 35 mm culture dish with suitable complete medium. According to the experiment, the culture dish was selected, and the area occupied reached more than 80% of the total area of the culture dish. The cells were incubated in 37 degrees Celsius box containing 5% CO2. When the cells were completely adherent, they could be transfected. 2H before transfection, 1.5 milliliters complete medium was replaced with the old medium. Cell sorting. Incubation. The mixture of this 400 ml was added into the cell culture medium of above monolayer, gently shaking the plate after mixing and keep it for incubation in 5% CO2 box. The mixing of core plasmid and packaging plasmid. Take a 1.5 milliliters sterile tube, and add 400 microsine L serum freed meme, 1.5 microsine G core plasmid and 1.5 microsine G virus packaging plasmid, 6 microsine L turbeffect, plasmid equals 2, 1, mixing them and keep them static for 15-20 min. Collecting virus supernatant. One. The cells are spread in 6-well plate or 12-well plate. The best density is 30% 70%. 2. For 6-well plate, 12 plate is half. 3. Add 2-5 grams/milliliters polybren. 4. After 12-24 H, change the fluid and do experiment. Virus infections. Infect mouse cells. 800 1600 microsine L virus plus 1600 minus 800 microsine L fresh medium equals 2.4 milliliters total. Infect human cells. 200 minus 800 microsine L virus plus 1200 microsine L minus 800 microsine L fresh medium equals 2 milliliters total. For 6-well plate, 12 plate is half.